right, everyone. I'm super glad to be here and be able to present what we do at DeepLife. So, in a nutshell, what we do, we accelerate drug discovery with AI-based digital twin of cells. So, I'm going to explain you a little bit what all these words means. So, first, maybe something that you heard again and again this past all across the day. So today, when you're looking at the drug discovery pipelines, it have like this V-shape, where between the first stage and the latest stage, when you define an hypothesis at the beginning, so you have about 10,000 of them, there's only one that will eventually reach the market. So you make that you have a success rate of 0.01%. It's very, very low. So today, in order to accelerate drug discovery, there is an urgent need to increase the biological reliability, the assets that we are building. So these types of problems of reliability is something that actually already existed in the past in another field, in mechanical engineering, actually. And what has been designed, like 40 years ago, is what we call digital twins, which are models that allow you to design and optimize a system before building it. And when you build the first prototype, you actually have a better understanding how the different pieces of your system are going to work together, how the action on one piece is going to impact another one. It's not a perfect model, but you have a good understanding of what's happening. So then the solution for developing new drugs would be to develop a digital twin of cells, right? Well, that's not as easy as it is. So in order to build a digital twin as something that will looks like what we have in mechanical engineering, actually we have to think a little bit differently. The main difference between biology and physics is that in biology we don't have laws, right? So we have to learn these laws from data. And hopefully today we have a lot of data, and we have also the algorithms to be able to learn that from data. So if you want an analogy, the types of model that we're building is, so we call it AI-based digital twins. So it's like teaching a robot to walk by showing him videos of human walking. So you're going to show your robot human walking, doing a lot of things, different pose, and he's going to learn the laws of physics, of mechanics, just by watching these videos, right? Once your system is trained, then you're going to ask him to do something that he never seen before, like, can you dance? And then he's going to dance. So what we're doing eventually is exactly the same thing, but applied on cells. So the types of data that we are getting is what we call omics data. So there's a bunch of them. There is genomics data, measuring the genome, epigenomic data, transcriptomics data, proteomics data. There's a bunch of these omics. And what these data are about is they are measuring the activity of the cells, of the different components of the cell very precisely. And today we have much more data that we got like in the history of humanity, where, for example, in our database, so we are mostly transcriptomic data today, but we have hundreds of millions of cells, and for each of these cells, we have the activity of all the genes that was taken from different patients. And then we can train the model to behave like a cell. Right, so the goal, we know that cells are not like unique and just isolated in their environment. There is actually the types of model that we're building is like a multi-level model where you have a modeling at the cell level, a modeling at the tissue level, an organism level. Today we're mostly focused on cell level, but the idea is really to integrate all these different levels to eventually have one model that allows us to answer like a bunch of questions, the first one being target identification. Right, so just presenting like few results. So just the first model that we built. So there is one question eventually is how do you validate that what your model is doing? So I heard like the previous talk was saying we should not trust generative models. But at some point, if you have a metric, like a benchmark, you can know if your model is completely hallucinating or not. So here what we did is what we call the retrospective analysis. So you try to make predictions on a disease that is known and what drugs or targets that actually made to the market. And then you look, okay, you can take method that exists in the state of the art, and then you take your method and you're looking at, based on the top targets that has been identified, which one of these targets are actually on the market. compared to methods that exist. And what we observe is that the method that we have today is tenfold better than what exists already in the state of the art. So I don't know if you're very familiar with digital twins, but actually when you're looking in mechanics, it's mainly a software. So it's there's companies that are selling software as a service. So you have a software what allow you to actually take a problem and design your, for example, if you want to design an engine, you're going to design your engine on this platform. So what we are building, so we're doing services, but also we are building like a software that tries to mimic exactly what you can have in the mechanical engineering, but for sales. So I will provide at the end of the presentation a small QR code, and we have also a free version you can test. But yeah, so the goal is to be like the digital twin of sales that you can use and kind of a self-service. Just a word about the team. So we are an international and interdisciplinary team. So we are like about 25. There is about 13 different nationalities in the company. So very like worldwide. Interdisciplinary, so people coming from physics, biology, AI, yeah, various expertise. And 90% of the company actually have a PhD. So really, what we do is really pushing the boundaries of AI applied to drug discovery. And that's it. So I think, perfect. We have some time for questions. And there's a QR code if you want to test the cell blueprint and look at our website. And we'll be very happy to take all your questions. Thank you.
Do you generate your own experimental data, or do you use only public data to do this?
Good question. Today, only public data, actually. But there is a lot of them, and I can give you the website where all the data are, and it's well known. The big problem is it's kind of the Far West. So the data, they don't have all the same format. So you need pipelines that are resilient enough to get all this data. So all the heterogeneity that you have at one hand, you need something very homogeneous at the other end. So that's the challenge. You have the challenge on the data, but also on the metadata. So sometimes they say, oh, this is this protocol and this is human. And you find out that it's not the right protocol and it's mouse, actually. So if you don't actually get this information also from the data, at some point you have like an ensemble of things that are super heterogeneous and you can't learn anything, actually. What you're learning is just the difference between the data sets. So that's what we put a lot of emphasis at the beginning of the company, actually.
So at the beginning, you were talking about video, looking at a video to learn the dynamic of something. But here, the data you use, I think it's like single-cell transcriptomic. It's like a snapshot, a photo, not a video. So do you infer like temporal dynamic from this?
Very good question also. So here, what we are building is what we can call a causal model, right? We are trying to predict how a cell is going to behave after perturbations. And the analogy with the robots, if you want like an even closer analogy, will be images. So you show images of human doing a lot of things, and that will be like your training data. So the main problem that you have when you do that, it's first that it's harder to generalize, right? But also it's observation data. So at some point what you want is a causal model. So you need actually an extra step which actually allow your model to come from observational to causal. And that's also like a big part of the development that we're doing internally. Yes?
And as you are in a multi-omic setup, how do you manage the curse of dimensionality regarding the performance of your model?
So at the end, indeed, the dimensionality is very high. So that's why we are using models that also like compress the information at some point. So we're trying to learn these embeddings on which all the different experimental data lies. And actually, one way to see if your model is like, if you you have like too much dimension based on the data that you have. If you have like 3 points and you try to draw like a line, at some point you will be able to draw something, right? But at the end, we have some benchmarks, as one I was showing, where you can see if your model is performing good or not. So the idea is that we're looking what's happening inside, but the real test of the fire is how you being better than what exists today that are based on other data and other priors. And that's how we know that the model has learned something that is useful for the task. So it's a bit different from what you can think about, like you building a system from element, so it's like we call bottom up, right? You try to understand all the mechanism and then you build a system and then you test it. Here we are really starting from the data, right? Trying to learn the system, it's more top down, and then we are looking if the system is performing well regarding the problem. And so if it doesn't learn like biology the way that we know it, it's not a problem because it's answering a question that we have, which is what is the right target for this problem? Then you have a question about interpretability that you can then derive. It's another problem. We're also working on that. But first, having a system that is better than what exists is very important. All right. Thank you.